Type 2 diabetes is a diet-induced neurological disorder. Hey everybody, in this video we're going to deep dive into type 2 diabetes, and I'm going to challenge the traditional thinking on this very common disease. It's very important for us to understand physiology comprehensively in the human body before we really understand the disease. In doing this, I am challenging the carbohydrate insulin model as well as the caloric intake model of diabetes. I'm presenting what really should be our primary understanding of what diabetes is and why it has the effects that it does in so many people. Thanks for joining us. Let's dive in. To start out, I want to demonstrate how insulin resistance can be induced in healthy people in a very short period of time without an excess of carbohydrates or calories. This study was in Cell Metabolism, a 2020 group out of Yale. They took healthy volunteers without any indications of metabolic disease, including insulin resistance, and they provided for these volunteers over 10 days a small glass of liquid, and the liquid contained a total of 75 calories, and it included sucralose as an artificial sweetener without any calories, and it also included 75 calories worth of maltodextrin. So this is a polymer, it's a synthetic polymer of glucose. And what they found is in just 75 calories in these drinks, a total of 7 drinks over 10 days, the daily average was 52 calories of this drink. Within 10 days, repeat testing showed insulin resistance, so increased fasting and postprandial insulin levels simply in this 10-day period without an excess of carbohydrates or total calories. Really, what this indicated in the study, that it wasn't sugar, it wasn't carbs, it wasn't calories. It was a mismatch between perceived calories, perceived by the brain, and actual calories. And it was this confusion that induced this state of insulin resistance. So I just want to start out with this shot over the bow, so to speak, with this really good study indicating that maybe we don't understand insulin resistance and diabetes clearly. To start out, we have to understand anatomy and some physiology to understand what I'm going to present today. So obvious diagram with the brain, you see the liver and the pancreas, and we will talk a lot about the vagus nerve here in yellow. This is the primary avenue for communication between the gut and the brain in both directions. Extremely important to understand these basic structures. There are signals from the gut to the brain. These are called afferent signals going from brain to gut. These are called efferent signals. And when I say gut, I'm really talking about liver, stomach, pancreas, intestines. But in this diagram, we really are just showing the liver and the pancreas because we're focusing on diabetes. Now, in that communication, that bidirectional communication between brain and the liver and in the pancreas, it's important to understand sort of two different systems or two different nerve systems. One is the vagus nerve, which I just mentioned. The vagus nerve carries signals in both directions, brain to gut, gut to brain. There's another system of nerves called the splanchnic nerves. This nerve system is what we call the paravertebral ganglia and nerves, so right by the spine. The splanchnic nerves largely carry sympathetic fibers. We'll talk more about that. Again, bidirectional, both directions, gut to brain, brain to gut, as does the vagus. And the vagus largely carries parasympathetic signals, and we'll talk more about that. So very basic anatomy of what we'll be talking about here, obviously, is the brain. An important center in the brain is called the hypothalamus. You see it there in blue. Another important structure for us today is the nucleus tractus solitarius. And this is sort of the gateway for signals coming from the gut into the brain. So it sort of filters and gates the signals coming from the afferent signals through the vagus nerve. Very important structure. We'll talk more about it. A little more detailed diagram of the brain and some of these important structures. You see there at the bottom, NTS, that's nucleus tractus solitarius, brainstem, and in the arcuate nucleus, there are a lot of different nuclei. In other words, clusters of cells that are sort of specialized. Maybe the important thing from this slide is just to understand there's a lot going on, a lot of brain centers. They all communicate with each other. It's incredibly complicated. But these are some of the very basic structures we need to understand in order to understand diabetes. Here's the idea, or the question: is type 2 diabetes a diet-induced neurological disease? And that's my argument today. Really, at the foundation of type 2 diabetes is a breakdown of the system of communication between the brain and the periphery, or specifically the gut, meaning liver, pancreas, fat, visceral fat, as well as the intestines. Again, structures like the hypothalamus, the medulla or the NTS, the dopamine system in the brain, signaling between those brain structures and those of the liver, pancreas, intestines, visceral fat. Neurological communication between these two centers that is really driving type 2 diabetes. Let's first talk about the traditional understanding, the traditional idea and theory about diabetes. This is an oversimplification, sure, but I want to make it representative. So classic understanding of type 2 diabetes is, you might call it overnutrition. People eat too much, or they have an excess of carbohydrates, too much sugar. People also talk about lipotoxicity, too much fat. Really what results is blood sugars that are too high. And because of these high blood sugars, or this increased glucose excursion from the intestines into the blood, you have increased or exaggerated insulin release from the pancreas in order to manage all that glucose that's being consumed. In all of that, there develops insulin resistance as a result of excess insulin. And that's one of the primary arguments, is that insulin resistance is caused fundamentally by too much insulin, and we're going to take issue with that. Others talk about visceral fat accumulating or storing fat in the abdomen, and that causing insulin resistance. Also take issue with that. And ultimately, this theory suggests that just chronic high demand for insulin from the pancreas causes dysfunction. It burns out. The beta cells of the pancreas are not able to keep up, and they end up becoming dysfunctional, can't keep up, and as a result, blood glucose increases, and that is technically when diabetes is typically diagnosed. Again, I submit that this understanding is incomplete and even incorrect. Let's talk about the basic hormone model of communication in the body. And this is something you learn in medical school or in a biological major in college, like biochemistry, which was my major. It's a very basic concept, and it's applied to almost every system. So in this model, you have an endocrine organ, say pancreas, or the thyroid gland, or the parathyroids, or the adrenal glands. These endocrine organs generate hormones like insulin or glucagon or thyroid hormone, cortisol. Under the appropriate conditions, that endocrine organ secretes that molecule, that hormone, into the blood. This hormone travels in the blood to a target organ. Say, for instance, in a case of insulin being secreted by the pancreas, it's secreted into the blood, goes through the bloodstream, and say the target organ is muscle, skeletal muscle. On that skeletal muscle, there are receptors specifically for that insulin, that hormone. And when insulin travels through the blood and activates that receptor in the muscle, it has its effect on the target organ or the muscle. So in other words, in this case, insulin would cause the muscle to take in glucose, for example, through its receptor. So this is the basic idea, or the classic hormone model. Endocrine organ creating a hormone, releasing that hormone into the blood. That hormone travels through the blood, arrives at a target organ, binds a receptor, is taken in, and has an ultimate effect of that target organ. Very basic model. In this diagram, just wanted to hit very basic physiology according to this traditional model of diabetes. So in here, we're looking at the intestines, and off to the left, upper left, you see an obvious diagram of the abdomen, the organs of the abdomen, and bottom right, you see those yellow cells. These are enterocytes, cells that line the intestine, and glucose is absorbed through those cells, and won't get into all the details, but ultimately through those cells, that glucose enters into the blood. So once in the intestines, the glucose passes into the bloodstream. It's entering into the venous blood, the veins of those intestines. The glucose travels through the veins, starting with the mesenteric veins and into the portal venous system, through the liver, up through the vena cava, hits the right side of the heart. From there it goes to the lungs. From the lungs it goes back to the left side of the heart, and the left ventricle of the heart, the main pump of the heart, pumps that now arterial blood through the arteries of the body, including the arteries that ultimately arrive at the pancreas. So from the intestines, there's this path of glucose through the venous blood to the heart, from the heart through the arterial blood to the pancreas. Another diagram of the pancreas there in blue, this represents what are called islets. These are the endocrine components of the pancreas, and the beta cells in those islets are the ones that generate insulin. So again, according to this traditional model, the glucose passes through the intestinal wall into the bloodstream, travels through the bloodstream to the pancreas, and binds to receptors in the beta cells of the islets of the pancreas, and that causes and stimulates the release of insulin from the pancreas. Now from there, this is where, according to this model, you have insulin released from the pancreas as a result of consuming food. And then the pancreas releases that insulin. That insulin goes into the bloodstream, travels through the bloodstream, again, venous, going to go into the pancreatic veins, the mesenteric veins, the portal vein through the liver, to the venous system in the heart, to the lungs, back to the heart, and then the heart pumps it through the arteries of the body to the peripheral tissues, in this instance, muscle, skeletal muscle. But I just wanted to understand what the physiology is for the actual passage of glucose from the intestines to the tissues. And in this traditional model of diabetes, this is what we're talking about. There's a breakdown of this system. So let's back up and look at the condition of diabetes. Let's look at it from an observational perspective. What do we see? Well, in type 2 diabetes, we see elevated levels of the following: glucose, leptin, GIP, insulin, GLP-1, now very famous. The injections, weight loss injections, are largely synthetic forms of GLP-1. But GLP-1 is elevated in diabetes, as is DPP-4. This is dipeptidyl peptidase 4. This is the enzyme, the molecule that breaks down GLP-1. That's at higher levels in the blood, as is prolactin, epinephrine, norepinephrine, glucagon, and amino acids. Now, all of those things really are, from high level perspective, those are things that go up after you eat in a healthy person. So all of those things are elevated fasting state in diabetics. Now, what you also see in diabetes is lower levels of ghrelin. Ghrelin is often called the hunger hormone. The general perception is that ghrelin is made in the stomach, and then when we don't eat, it goes up and travels through the blood and stimulates those centers in the brain that cause or stimulate hunger. But it's interesting, what you see is a flip. You have high levels of all of these molecules up top, and the bottom hunger hormone is actually lower than normal in diabetes. And this is really important. So when we recognize this, we have to back up and realize, okay, something is off with the whole system. There's almost a global dysfunction represented by these alterations in fasting serum of diabetics. To understand diabetes and to understand what I'm presenting today, a person has to understand the autonomic nervous system. You can think of the autonomic nervous system as the automatic nervous system. In other words, big part of the nervous system that manages and controls everything that's involuntary, things you never think about. Dilation of the pupil, pulse, blood pressure, digestion, bladder control, all kinds of things going on below the level of consciousness. Well, it's largely this autonomic nervous system that's managing all of this. Well, there are two branches of the autonomic nervous system. One is called the sympathetic nervous system. This is known as the fight or flight branch of the autonomic nervous system. You're out in the woods hiking with your kids, you see a grizzly bear, boom, that adrenaline hits you, and there's hundreds of different changes that happen in your body quite quickly to take care of that situation. And so this is really a reflection of activity of the sympathetic nervous system. In that scenario, seeing the bear, sympathetic activity goes way up. Now, in antagonism to the sympathetic system, or sort of opposite, is the parasympathetic nervous system. This is also known as rest and digest, recovery, healing. Very important. Both of these branches of the autonomic nervous system are incredibly important in a healthy person, a person who is metabolically healthy. Those two systems have to be in balance. In human physiology, so much involves cycling, meaning swinging from one extreme to the other, or cycling between two different states. You think of the wake and sleep cycle, based largely on the sun. Well, just about everything in the body is involved in some type of cycle, and they almost all involve fluctuations in the balance of this autonomic nervous system. Exercise, sleep, eating. We exercise, we get more sympathetic activity. After exercise, when we are recovering, in a healthy person, you're going to get more parasympathetic. And wake and sleep cycles, same thing, as well as eating. So in a healthy human being without metabolic disease, these two systems, sympathetic nervous system and the parasympathetic nervous system, are both active, but you see consistent fluctuations in different physiological states. So, what do we see in this autonomic nervous system in type 2 diabetes? What we see is a chronically elevated sympathetic activity and a chronically low activity in the parasympathetic. Now, you have to understand heart rate variability, or HRV. This is, you can think of it as a measure of flexibility in the autonomic nervous system, specifically dealing with the heart rate. As a person goes from sitting to standing, you want your heart rate to pick up a little bit to keep up with the need for blood pressure in a different position. So this heart rate variability is something that is very helpful in measuring balance in the autonomic nervous system. And it's also a direct result of vagus nerve afferent health. You go back to that vagus nerve and its communication between the brain and the gut, meaning liver, pancreas, and other organs of the abdomen. The afferent signals, those are signals going from gut up to brain. In diabetes, what we see is a decreased afferent signaling from gut to the brain. This is uniformly the case, and again, it reflects dysfunction in that vagus nerve. So a little bit on the carbohydrate insulin model, or the energy balance model, and by that I really mean calories in, calories out. Imagine you're a store owner. You own a shoe store in a city, and you come in one day to your shop and you notice the front window of your shop is broken, and on the floor there's a brick or a couple of bricks. Very frustrating, aggravating, it's loss, it's problem. You conclude these bricks did this. The bricks broke my window, and in one sense, that's true. A month later, after getting it repaired, you come in and there's a hammer on your store floor and the front window is broken again. This time you say, this is the hammer. The hammer did this. With this analogy, what I'm saying is that with the carbohydrate insulin model, as well as the energy balance model, again, calories in, calories out model, it's too simplified. It's too reductionistic. It's oversimplified. Neither of them are complete or whole. They're very inadequate. And because of that, I would say they're wrong. This is a very important graph. When I first saw this graph in a paper by Young's Bruning group from the Max Planck Institute, I thought, hallelujah. I'm a nerd, I know, but I saw this graph and it represents the different signaling between gut and brain and timing of signals. And I'll explain more about that. But this is a very important graph for us to understand diabetes and metabolic disease. And the traditional thinking about diabetes is based on the hormonal model of signaling and communication. And of those three waves in this graph, the first one, meaning the one furthest to the left, that represents what we'll talk about more, and that is sensory perception, external signals to the brain. The middle one are signals, neurological signals between the gut and the brain. And that last one furthest to the right that says homeostatic hormonal regulation, this is the hormones in the body communicating between gut and brain. So again, the traditional idea or teaching about diabetes is really focused or based in the third time frame, the third stage of communication. It's behind. It is not acknowledging communication that is happening much quicker than that. I'll go back to that. So as an analogy, suppose I am in Minneapolis, Minnesota, and I want to communicate with you, and you are down in New Orleans. I think that's probably 1,500 miles, don't know for sure. And I consider, okay, how can I communicate with my friend Bob, who's down in New Orleans? I got some options. First one, I can go find a bottle. I write out my message to Bob on a piece of paper, roll up the paper, put it in the bottle, put the cap on the bottle, throw it in the Mississippi River tributary system. Eventually, hopefully that bottle is going to reach Bob in New Orleans. He gets it, he finds it, he opens the bottle and gets the message. That's one option. Second option, classic, is to use the postal system, U.S. postal system. So I write my message down, put it in an envelope, put the address in on the envelope for my friend Bob in New Orleans, put it in the mailbox, off it goes. It's going to get to my friend Bob in New Orleans. But wait a minute, we live in 2024, we've got satellites, we've got digital technology everywhere. I have a cell phone in my palm. I can text Bob. And assuming he's got a phone in his hand, I can send a signal to Bob extremely quickly, much faster than either the letter in the mail or the bottle in the river. So there's a major point here. This comparison is important to understand that there's a lot going on in a person's body, specifically communication between brain and gut to brain, before hormones have their effect dealing with insulin and with glucose. That's the point. Okay, so let's talk more about this neurological communication through nerves between brain and gut, gut to brain. Say I walk in the door and I smell dinner, smells delicious. I walk in the kitchen, I see whatever, I see steaks or lasagna or something. I can smell it, I see it. I walk in, I smell that steak or that lasagna, whatever it is. I smell it, I see it, and my brain perceives those signals. I haven't touched the food yet. The brain perceives it and processes it in a way to prepare my body to digest that. Incredibly important, incredibly fast. And this happens much quicker than the signals that result from the food actually being in my stomach or intestines and the nutrients being in my blood. It happens much faster. That's called exteroception. External signals being perceived by the brain making a prediction on what's contained in that food based on smell, visual, and my memory to prepare my body to most effectively and efficiently digest and metabolize that food that I'm going to be eating. Now, I sit down, I eat that steak, I chew it up, starting to digest, it enters the intestines, and it brushes against enteroendocrine cells on the lining of the intestines. Those enteroendocrine cells can recognize through receptors what it is exactly that I just ate. At that point, those enteroendocrine cells signal through the vagus nerve, through those afferent signal systems to the brain, and that communication from the layer of the intestine to the brain takes about 3 milliseconds. Very fast. So as part of that meal, say I had a piece of bread and that glucose is now in the intestine. It brushes up against those sensory cells, enteroendocrine cells, and say it enters the blood at that point, travels through the veins, to the arteries, to the pancreas, and once the glucose hits the pancreas, those beta cells of the islets stimulates insulin secretion from the pancreas at that point. There's another method of signaling, and this is much longer term. A lot of people are familiar with leptin. Leptin's created by actually a lot of cell types. The common thought is that it's created by fat cells, adipocytes. It's actually created in a lot of other tissues also, and that's very important. But the classic idea is that, hey, fat cells make leptin, and leptin in the blood travels to the brain and signals to the brain how much storage there is, how much fuel is stored as fat in the body. This is a much longer term method of communicating between the fuel storage sites of the body and the brain. So again, here's this graph from a 2022 paper. The first wave is representing the exteroceptive signals, vision, smell. This happens very quickly. And it is at that perception where things start to change from a metabolic standpoint. Then when the food is actually in the intestines, it signals to the brain. That's the second wave, and that happens through the vagus nerve. And then the third and slowest or later wave is the hormonal signals that occur. In this slide, you see a comparison, a time comparison between neurological communication and hormonal communication. So nerve speed versus hormone speed. On the left, say you first encounter food, you smell it, you see it very quickly, meaning in a minute or two, there are changes that happen in the hypothalamus. There are some different clusters of cells, POMC, and that stands for pro-opiomelanocortin neurons. There's another cluster of neurons in the arcuate nucleus of the hypothalamus called Gaudy-related peptide neuron. These cell or neuron populations in this part of the brain, they're again antagonistic with each other. When we're hungry, we haven't eaten, the Gaudy-related peptide neurons, they're active. They're driving the sense of hunger. And in that state of hunger and not eating, the POMC neurons are inactive. When we see, when we smell food, the balance between those two neuron clusters changes very rapidly within a minute or two. The Gaudy-related peptide neurons are suppressed, they're less active, and the POMC neurons become very active. And it's very soon after that, because of that change in the brain, and signaling through both the sympathetic splanchnic fibers as well as the vagus efferent fibers, stimulates insulin secretion from the pancreas. This is before any food has been ingested. There's a pulse of insulin released from the pancreas before anything has entered the mouth or intestines or any glucose has entered the blood or arrived at the beta cells of the pancreas. But it's not just the pancreas that is altered in physiology with these visual and olfactory signals. It's the liver. These same neurological signals activate a very important complex called mTOR XBP1, which is preparing the hepatocytes, the cells of the liver, to prepare for stress that's associated with metabolizing processing the food that's eaten. These are changes that occur in these organs very quickly before any food has been ingested. I would add adipocytes, the fat cells, brown fat as well as white fat. There are signals sent through the nerves from the brain to fat cells as a result of seeing and smelling food. All of this happens really within 5 to 8 minutes in most studies. So all that is happening before we've eaten anything. We put the food in our mouth, it goes from our mouth to the stomach, and most studies indicate that the food remains in the stomach for about an hour. Depends on what it is we're eating, but that's a pretty decent estimation. Is the food is churned, it's broken down by some basic enzymes in the stomach, and after an hour, maybe hour and a half, the food starts leaving the stomach and enters the small intestine. So 60 minutes, 90 minutes after actually eating, that is when the glucose in the blood peaks, and when insulin really, after a meal, is peaking as well. So you see this very important and very profound difference in timing of these communication in these two systems. Nerves communicate much faster. It's almost like precognition in that Tom Cruise movie Minority Report. It's before food is ingested and received. The brain makes a prediction of what's in it, and it starts all of these metabolic changes before digestion has started. It's worth mentioning that those 3 waves of communication, they really all involve the vagus nerve, both afferent and efferent, up and down that vagus nerve. All of these modes of signaling involve that important vagus nerve, and I would speculate probably also the splanchnic nerves, both afferent and efferent. Let's back up and start again. Ask the question, well, we're talking about diabetes, type 2 diabetes. What stimulates insulin release from the beta cells of the pancreas? Basic question. Really, there are 3 phases of insulin release. All of these phases of insulin release are pulsatile in nature. In other words, insulin release from the pancreas is always occurring in a pulsatile manner. Again, back to that cycling idea. We have basal insulin secretion. This is just the insulin that's being released from the pancreas when we haven't eaten. We still have some insulin in our blood, and a lot of doctors, me included, will measure fasting insulin levels to get an idea of where a person is as far as their insulin sensitivity. Then there's the cephalic phase. And this is what we just got done talking about. These are the cephalic phase. Cephalic, the meaning of that word really is head. This is the phase of insulin release that occurs before the pancreas sees any glucose. This is based on exteroceptive signals, visual, olfactory. And then there's the post-ingestive or post-ingestion phase. This is actual glucose, amino acids, fatty acids stimulating insulin release from the pancreas based on their arriving at the pancreas. Okay, cephalic phase insulin secretion. There's an important structure in the brain called the insular cortex. And this is sort of the clearinghouse. It's a processing center, a center of integration of a lot of signals, both exteroceptive, outside signals, as well as interoceptive signals, interstate signals from the gut and other places in the body. The insular cortex is incredibly important at receiving all those signals and making sense of them. And it's very important in this process of the brain making a prediction about what is in food based on vision and smell and memory. Insular cortex, very important. And of course, because we're basing that prediction on memory or previous exposures to foods, it involves the hippocampus of the brain. A lot of centers in the brain involved in all of this. It's an incredible system. But this, again, as I mentioned, results in an almost instant change in balance in the hypothalamus, in that arcuate nucleus between the POMC neurons and the Gaudy-related peptide neurons. And that results in efferent signals from the hypothalamus to both the alpha cells and the beta cells of the pancreas. There are some studies that suggest that for the cephalic phase insulin release, the alpha cells are required, and of course the beta cells are required because they're the cells that generate insulin. So really there's a symphony of things happening, neurotransmitters that are all interconnected and achieving a balance in our bodies for effective and efficient metabolism and ingestion of food. So the cephalic phase, again, we haven't ingested or digested any food. This cephalic phase of insulin release involves the liver, the pancreas, adipocytes, the stomach, the brain. This is a slide representing the insular cortex, and it's based on visual, olfactory, and taste, gustatory. We taste something that adds additional information for the brain. None of those nutrients have arrived at the pancreas or the other organs, but it's more information to base a prediction upon. This is a slide I put in just to demonstrate a principle, and that principle is that human beings, we love to simplify things because it helps us think that we understand. We're all reductionists. We all oversimplify things for this reason, and maybe we have to, but this is a slide in upper right. This is just looking at the insular cortex and the interconnected neurons in the insular cortex and how they're processing and interpreting different signals. Incredibly complicated. Talk about a sophisticated, advanced supercomputer. So there are, functional MRIs have changed the way we understand neurobiology pretty dramatically over the last 20 years, including in diabetes. And plenty of studies, I won't go into them, but they show differences in both structure of these centers of the brain as well as physiology. In other words, the brains of folks who have diabetes and are obese, they look different, they function different in these key centers, and it's important to note that. So again, revisiting this slide, this idea that diabetes type 2 fundamentally is resulting from a breakdown of these systems of communication between the gut and the brain. Nerve communication breaks down. Very important system. Another diagram showing elements of the hypothalamus. Again, everything in this presentation is oversimplified, but in that arcuate nucleus, you've got the POMC neurons, a Gowdy-related peptide. There's also a cluster of cells of neurons called the PNOC neurons. They're also incredibly important in all of this, as well as other oxytocin-based neurons. All these different neuron clusters in the brain are involved in metabolism, in insulin release, and they're interacting with each other in an incredibly complicated way. As part of the cephalic phase, I mentioned changes that happen in the liver. Very important changes in the liver. Again, when these POMC neurons and the Gowdy-related peptide neurons, when they sort of flip in their balance, this results in signals, neurological signals being sent through the vagus efferent nerve fibers from the brain to the liver. And those signals stimulate changes in the liver cells, including transcription of specific genes that are involved in managing stress involved with metabolizing and ingesting food, and in prepping the liver for all of this. These signals also control insulin sensitivity in the liver. Again, you can keep everything static. Blood glucose is the same, blood insulin levels, peripheral insulin sensitivity is unchanged, but if you change the activity in these two cell populations in the arcuate nucleus, you get changes in the liver's sensitivity to insulin. An example of peripheral insulin sensitivity being controlled in the brain, in the hypothalamus. Amazing study. You see the reference there. So that's the liver. Let's look at the pancreas. The same type of communications going on between centers of the brain, including the hypothalamus and the pancreas, both afferent and efferent. And this just demonstrates how complicated it is. So in this slide, you just see an example or a model of the beta cell. The beta cell, again, is the cell that releases insulin under those conditions, the right conditions. And these are just representations of some of the molecules, some of the neurotransmitters involved in regulating when and how much insulin is released from the pancreas. Very sophisticated, and we oversimplify it. So what do we see in cephalic phase insulin release? It happens very quickly. Within usually 2 minutes, the pancreas releases a pulse of insulin into the blood before any glucose or amino acids have arrived at the pancreas. This is a great study, old study, and it's looking at the effect of cutting the vagus nerve, what that does to cephalic phase insulin secretion in sheep. That was their model. And what they found is that cephalic phase insulin release, that pulse of insulin from the pancreas, if you cut the vagus nerve, it goes away. You don't get that pulse of insulin released in that cephalic phase from the pancreas. Bottom line with this study is they demonstrated that in order to have that cephalic phase insulin release, you have to have an active functional vagus nerve. Here's another study, and they looked at what happens when you get rid of that cephalic phase insulin release. In this study, this was in human subjects, diabetes subjects compared to controls, non-diabetics. What they found is that in diabetic people, they didn't have a cephalic phase insulin release. And so what they did in the diabetic patients is they gave an injection, some exogenous insulin that was representative of the typical release from the pancreas in that cephalic phase. And what they found is that giving these diabetic patients a pulse of insulin where the cephalic phase insulin release should occur, that changed blood glucose area under the curve. It changed insulin area under the curve. In other words, that pulse of insulin at the right time improved dramatically the glucose homeostasis in diabetic individuals. Addition of this insulin in the cephalic phase corrected glucose excursion to near normal levels. Again, these are diabetic patients who are demonstrating near normal physiology when that insulin is given at the right time in that cephalic phase. There's a number of rabbit holes that you can go down, and looking at, well, the cephalic phase insulin release, what is all involved in that. There's a lot of different systems in the body involved in it, neurological systems. Here's a paper that showed that in the hypothalamus, there's a specific molecule, interleukin-1 beta, very commonly recognized cytokine, but they found that for insulin release in the cephalic phase, it required production and release of this interleukin-1 beta in the hypothalamus. If you block that interleukin-1 beta in the hypothalamus, you don't get your cephalic phase insulin release. In this study in American Diabetes Association Journal, this is human subjects. What they determined is that cephalic phase insulin release from the pancreas, that's absolutely required for normal blood glucose and insulin levels after a meal. That cephalic phase was dependent on the vagus nerve, both cholinergic and non-cholinergic. In other words, it required the neurotransmitter acetylcholine as well as other neurotransmitters in order to function properly. When you block those neurotransmitters, normal people without diabetes, their blood glucose after meal is elevated, as is their insulin. In other words, cephalic phase insulin secretion, it involves the vagus nerve, and it's very important for normal glucose and pancreatic physiology. You can look at the other side of that. This is in a rodent model. There's a common model of diabetes in rodents. The rodents are given a chemical, STZ there, and it really damages the beta cells. And it knocks down their ability to generate and secrete insulin. And it's a great model for type 1 diabetes, but also type 2 diabetes, later stage type 2 diabetes, when the pancreas can't compensate anymore. Blood glucose goes up. What these researchers found in this rodent model is in that setting, after these rodents had been given essentially this poison that largely knocks out beta cells, if they stimulated the vagus nerve, in other words, with electrodes, they stimulated the vagus nerve, what they found is that pancreatic beta cell function was preserved to a significant degree, as well as insulin secretion. So even in this model of beta cell loss and apoptosis, that stimulation of the vagus nerve helped to compensate for that as far as glucose and insulin release from the pancreas. There's a very important point I want to make, and that is that so much of our conversation today, talking about obesity and diabetes, we talk about hormones. Again, and it is represented in that graph, that three waves of signaling. We're stuck in this model of hormones, and it even includes insulin. So insulin, we know it is a hormone, but it also is a neurotransmitter. So in this study, they found that insulin release from the beta cells of the pancreas, that insulin binds to vagal afferent fibers, insulin receptors on the vagus nerve, right there in the islet. Insulin release from the pancreas binds those insulin receptors on the vagus afferent fibers and signals to the brain through the NTS, back up to the brain and those brain centers. When you knock that out, you knock out that communication from pancreas through the vagus nerve using insulin, you get obesity. These rodents, this is a rodent model, they became hyperphagic. They ate a lot. They could not achieve satisfaction like normal mice. So again, we have to rethink these systems and question our understanding. Insulin is a hormone. Is it a neurotransmitter also? Bottom line, those insulin signals from the pancreas also signal to the brain through the vagus nerve through insulin receptors. And here's a diagram representing that. You can see the pancreas on the right, the brain on the left, the nodose ganglion that's up in the base of the brain essentially, and all the vagal afferent fiber signals are going through there and the islet, that sort of cluster in the middle. You see vagal fibers there. And so insulin release there signals to the brain through those vagal afferent fibers. In the other direction, here's another distinct hypothalamus to pancreas circuit that modulates insulin secretion. This center in the hypothalamus, it's in the paraventricular nucleus, and it uses oxytocin. We typically think of oxytocin in the setting of breastfeeding or birthing or connection, emotional connection with other people. Again, these things they're neurotransmitters, and we tend to oversimplify and think of them only in certain contexts. Oxytocin, it's a neurotransmitter, very important in normal cell signaling, insulin signaling. It suppresses insulin secretion. Here's another study out of Cell Reports based in the hypothalamus of the brain. Just another element of regulation of insulin secretion and blood glucose. Leptin, again, we think of it as a hormone released by fat cells. It's a neurotransmitter. There are leptin receptors on the vagus nerve. Leptin's made in the brain. There are leptin receptors in the brain. This study demonstrated, interesting, in the setting of insulin resistance, hyperglycemia, if you inject leptin into a particular part of the hypothalamus, it corrects blood glucose and insulin sensitivity for a certain period of time. Haven't changed anything else in the body. You inject leptin into a certain part of the brain, it corrects insulin resistance and blood glucose. And so there's this fascinating interplay between insulin in the brain as well as leptin. All very important and an important piece of the puzzle. This is just such a fascinating concept that peripheral insulin sensitivity is controlled to some degree in the brain. Almost remotely, these centers in the brain are controlling how sensitive peripheral tissues are to the effects of insulin. And in this study, they looked at Gaudy-related neurons, again, central to our conversation. Activity in those neurons alters and affects insulin sensitivity in brown adipose cells. This brown fat. This fat is common in babies. Babies can't shiver, so they have a lot of it to keep them warm. This type of fat is just burning through fuel and generating heat, and it's largely controlled by the sympathetic nervous system. And what these researchers showed is that in the hypothalamus, the Gaudy-related peptide neurons, to some degree, they're controlling insulin sensitivity peripherally through their connection and relationship and communication with brown adipocytes. Central control of peripheral insulin sensitivity. So not just controlling how much insulin is released from the pancreas, but how sensitive peripheral tissues are to the insulin that's released from the pancreas. I mentioned PNOC cells previously in the arcuate nucleus. This is an additional population or cluster of neurons that are very important. These cells, the PNOC cells, they suppress the POMC cell activity neurons. And so when the PNOC neurons are active, they shift the balance between POMC neurons and Gaudy-related peptide neurons in the direction of hunger and food seeking. Fascinating. And then so you can look at high fat diet. This is a rodent model. High fat in these studies means high fat, high sugar. What they found is that when those rodents consumed high fat, high sugar or hyperpalatable foods, it activated the PNOC cells and resulted in suppressed activity of the POMC neurons. So essentially, this is one of many circuits or pathways by which high fat, high sugar foods suppress satisfaction. They induce this state of eating for a prolonged period for a meal and food seeking. These PNOC cells use GABA, very common neurotransmitter. They release GABA at their interface with these POMC neurons, and in that way inhibit those POMC neurons. I thought I would throw this one in there. Glial cells. So in the brain there are different cell populations, neurons. It's about 80 to 100 billion neurons. There's about 5 times as many what we call glial cells, supportive cells. And these other glial cells are also involved in metabolism, weight maintenance, inflammation, all kinds of processes. And in this study from Cell Metabolism, they demonstrate that some of these microglial populations, they're involved in regulation of appetite, eating, preference for high-fat foods, and it involves UCP2, which is uncoupling protein involved in fat metabolism. So according to this study, eating a high-fat diet, high fat, high sugar, resulted in inflammation in these microglial cells. And this cell population and this protein were necessary for peripheral inflammation, obesity. Incredible how many systems are involved, all these neuronal populations and how they are integrated and communicate with each other. Just want to revisit this slide. Diabetes is primarily a neurological disease. It is a diet-induced breakdown of communication, neurological communication between the brain and the gut through a couple of different nerve populations, vagal populations as well as sympathetic and splanchnic nerves. These following slides demonstrate some of the anatomy between pancreas and the brain. And you see different parts of this nerve system, a lot of different structures involved. And in there in that box, you see sensory mediators: trypsin, cholecystokinin, secretin, serotonin, histamine, bradykinin, vasopressin inhibitory peptide, BDNF, prostaglandin, interleukin-1b, ATP, heat, pressure. These are all of the molecules involved in this communication between gut and brain. I just want to roll through here. This is specifically for afferent signaling. So in other words, pancreas communicating with the brain. It involves all of these molecules through these nerve populations, gut to brain. Here's a representation of the efferent, so brain to gut. Little different molecules used, as well as fibers. Also very complicated, multiple steps to it. Happens very quickly. So in this slide from this review article, another way where the pancreas communicates with the brain, and the point of this was, in this diagram, it's indicating or suggesting that all of these molecules are traveling through the blood there in red and communicate through the blood to the brain. But if you look at each one of those molecules in that list, glucose, insulin, glucagon, leptin, ghrelin, adipinectin, amylin, cholecystokinin, GLP-1, you get the idea. All of those have receptors on the vagus nerve. So again, we have to step back and think, wait a minute, that three-wave graph, yes, they travel through the blood, but they're communicating in a much more fast and efficient way through nerves. All of those things are also generated in the brain. Should indicate to us it's a far more complicated system than we understand. These are not necessarily hormones alone. They're probably primarily neurotransmitters. Within the islet of the pancreas, you see in this diagram just a very simplified representation of the alpha cells, the beta cells, and there's a lot going on. And between the nerves, different neurotransmitters, alpha cells, beta cells regulating each other, tons going on. We love to simplify. Vagus nerve. It is the main and primary mediator between signaling between the brain and the gut. There are several studies showing the effect of diet and food on the vagus nerve. In this study from Scientific Reports, this was in rats, they showed that a high carbohydrate diet as well as a high fat diet, they both impair vagus nerve signaling, that is afferent signaling up from the gut to the brain. Both of those macronutrients at high levels suppress afferent signaling through the vagus nerve and tended to be additive. You add those two macronutrients together, the effect on the vagus nerve afferent fibers was even greater. This is a study out of the Florida group released less than a month ago, showing that there are fibers, vagal afferent fibers, that convey sensory signaling for carbohydrates, sugar. There are separate vagal afferent fibers that convey the messaging of fats, and that these two different fiber types can result in overeating. You combine them, you activate both of those vagal afferent fibers, you get additive effects. What you see in these rodents is very much a promotion of eating behavior. They ate more. This is from the Journal of Physiology, a little older article. This was in mice, and what they showed, same thing. High fat, high sugar diet suppresses activity of the vagus afferent fibers. This was done for both mechanoreception, in other words, stretch. When the stomach or the intestines are full or filled with food, they stretch, and that's mechanoreceptor mediated signals through the vagus nerve. You can also have chemosensation. Again, like the previous article, sugar, fat, those macronutrients stimulate chemoreceptors, whereas stretch, those activate mechanoreceptors. Well, it turns out that high fat, high sugar diet suppresses all the above signaling of chemoreceptors in the form of sugar and fat, as well as mechanoreceptors, stretch receptors for stomach and intestine. This was in mice. They started them on the high-fat, high-sugar diet at 4 weeks and fed them accordingly to 14 weeks. You see there in the graph pretty significant changes in vagus nerve activity. This study out of Scientific Reports, same idea. High-fat, high-sugar diet. They took this one step further and they looked at HRV and sympathetic activity and autonomic balance. And they showed, in the quote there, reduced parasympathetic output contributes to early high-fat diet-induced heart rate dysregulation. So they're looking at the heart rate and heart rate variability really is governed between this balance between sympathetic and parasympathetic. And so they took it one step further. They showed that in this high-fat, high-sugar diet, you had changes in the heart rate and heart rate intervals, and it was really as a result of suppressed parasympathetic activity, again, pointing to a decrease in vagal afferent signaling. So let's back up. Again, basic idea. We're talking about diabetes, brain to pancreas. In this diagram, once again, we have afferent arrows or signaling from pancreas to the brain. Efferent is brain to pancreas. And high-fat, high-sugar diet, what we see with exposure to that type of food, you get a degeneration of the afferent, vagal afferent signals. Ultimately, what this translates into is in that autonomic nervous system, there's far more sympathetic activity relative to parasympathetic activity. Accordingly, HRV decreases. So here's a study. They looked at heart rate variability in type 2 diabetes specifically. And this was a meta-analysis, a great study. What they showed essentially uniformly is in diabetes, HRV is low. Not a surprise, again, reviewing this physiology. It's not controversial. It's very widely accepted. This study, what they found is that HRV decreases before a person has diabetes. And in the summary there, they say these findings support the concept that cardiac autonomic dysfunction, code for low HRV, precedes the clinical diagnosis of type 2 diabetes. Again, some people will say, hey, look, with diabetes, high blood glucose, hyperglycemia, that affects nerves, and that the change in nerve activity and physiology, that's a result of diabetes and high blood glucose. Not so. Several other studies like this show that you see this change in HRV, this change in parasympathetic activity and sympathetic activity before you get prediabetes and diabetes. This would suggest that more than a result of diabetes, this is a driver of it. Again, neurological basis of diabetes. This is from the journal Circulation. What they're looking at is they're comparing what's called MSNA, muscle sympathetic nerve activity, as a general measure of sympathetic activity. Again, it's relative, and as the vagal afferent signaling goes down, HRV goes down, and we get this imbalance, more sympathetic compared to parasympathetic. In this article, it looks at muscle sympathetic nerve activity in normal controls in people with high blood pressure and in people with diabetes and high blood pressure. And as you can see, there's sort of a progression. Compared to normal controls, people who have high blood pressure have somewhat higher sympathetic activity. People who have diabetes have even higher sympathetic activity. If people have both diabetes and hypertension, significantly higher still MSNA, muscle sympathetic nerve activity. Again, the idea is we're shifting that autonomic nervous system further and further into a sympathetic, hyperactive state. Here's a light bulb. I think this is a very effective analogy. Think of a light bulb, and the light bulb has a filament, and you turn the light on, and there's a current, electrical current that goes through that filament, and it lights off. It emits energy, and we perceive it as light. Well, if you put too much of a current through that filament, or if you leave it on for too long, that light on, that filament burns out and it no longer works. It actually breaks that circuit and the current can no longer go through there. This is a wonderful analogy for what's going on in the autonomic nervous system. With the shift to the sympathetic hyperactivity, again, it's like pedal to the metal, chronic high activity in the sympathetic system. And what you see is sort of a J curve. Initially, there's a burst of sympathetic activity in end organs. And over time, you get failure of the connection between the sympathetic nerves and the end organ. In this case, we're talking about the pancreas. We'll talk about the liver too. But over time, there's degeneration of those sympathetic nerves and sympathetic signaling, just like that filament in the light bulb. Very good analogy. And this is a study, I want to say a couple of years ago, in Science Advances. This is from a radiology group. And they have a genius way of imaging the sympathetic nerves in the liver. And what they did, this is 3D imaging of all the sympathetic fibers in liver, and they followed, both in rodent and human models, the progression of non-alcoholic fatty liver disease and into NASH, non-alcoholic steatohepatitis. Sort of a progression from fatty liver into inflamed liver and cirrhosis. And this is precisely what they describe. They saw parallel signs of mild degeneration and axonal sprouting of sympathetic innervations in early stages of experimental NAFLD, and a collapse of sympathetic arborization in steatohepatitis. Again, there's that initial increase of sympathetic activity, and over time, there's a collapse in the fibers and in the activity. And they state that chronic sympathetic hyperexcitation is a key factor in that axonal degeneration. And here from the same study, they also quantified noradrenaline, the primary neurotransmitter of the sympathetic nerve fibers, as well as galenin, which is like neuropeptide Y, sort of co-secreted with the noradrenaline. And what they found there in K, L, and M, compared to controls, subjects with progressive NAFLD and NASH showed lower activity of not just the sympathetic nerves, but the neurotransmitter, noradrenaline and galenin. Using the same concept, let's look at the pancreas. We actually see the same thing. This is in humans. Similar technology. They are creating a 3D atlas of the nerves in general innervating the pancreas in controls and in diabetes. What they saw is what they said islet nerve density. Again, the islets, they contain alpha cells, beta cells. Islet nerve density was increased in non-obese diabetic mice, in mice treated with that STZ, and in pancreata of human donors with type 2 diabetes. So they looked at human beings who donated organs, and they compared the non-diabetic organ donors to the diabetic organ donors. And what they found is in the diabetes people, individuals, donors, there was a greater nerve density around the islets. Rings a bell. Is it like the liver where you have this expansion of sympathetic fibers? Another similar study, and this is looking at sympathetic innervation, sympathetic fibers in type 2 diabetes human beings. And what they found is a decrease in sympathetic fibers in the islets of diabetic patients. And so I would speculate here that these two different studies, one showing increased nerve density, one showing decreased nerve density, makes sense that they're at different time frames of that light bulb physiology, with that initial sympathetic increase in activity followed by the collapse. In the study, they also showed imaging, CT scanning, PET scan, and here on the right, this is specifically, they're scanning using labeled tracers for noradrenaline. And what you can see there and see the right upper image there, and this is a cross section of a human being, and you can see the liver there as well as the pancreas. That's normal, and the one below it is in a diabetic. And what you see is a significantly and obviously decreased level of signal for sympathetic nerve fibers in the diabetic pancreas, consistent with this idea of a light bulb and burning out. If we back up, we've talked about sympathetic nerves, parasympathetic nerves in the setting of diabetes. Now, if you intervene early in that process of that neurological change, this was in a mouse model. What they did, and this is type 1 diabetic mice, again, treated with STZ to knock out those beta cells. What they did is they cut the sympathetic nerve fibers. And what they saw is rodents did not progress into type 1 diabetes. The controls in whom they had a sham operation, you didn't cut those sympathetic nerves, they went on to develop type 1 diabetes. If you cut the sympathetic nerves, it stopped the progression. Again, this argues that sympathetic nerves and nerves of the autonomic nervous system have a very important primary role in the pathophysiology of diabetes. Sort of further down the road, say you have some of that collapse of the sympathetic fiber activity in the setting of type 2 diabetes. You have now decreased sympathetic activity. So in this model, this paper, in the setting of diabetes, so pre and diagnosed diabetes, instead of the sympathetic nerve fibers, they stimulated vagus efferent fibers, parasympathetic fibers going from brain to the pancreas and to the liver. And what they saw is that blood glucose and insulin secretion improved. This is in diabetics. You stimulate the vagus nerve, it improved their diabetes, sort of the inverse of that last study. This is from the same paper. It's a diagram of the vagus nerve, 4 different samples of it, and you see degeneration of the vagal nerve fibers as the diabetes progresses. And again, they show that even when a person starts developing insulin resistance, they already have some degeneration of the vagal afferent fibers. So I want to back up and explain something. You can go and you can look at all the organs that are involved in metabolic disease: heart, lungs, liver, pancreas, kidneys, fat, intestine. And what you see as the global autonomic function declines and you get the shift to a greater sympathetic activity and low parasympathetic activity, that imbalance affects every organ, and I would suggest drives end organ dysfunction. So in the case of the pancreas, we see that. It also drives hypertension, dyslipidemia, coronary artery disease. This has been done in rodent models. You look at the coronary arteries, and in models of atherosclerosis, where those plaques are, you have an expansion of those sympathetic nerve fibers just adjacent right by where those plaques are. Very consistent with this. And also in those rodent models, what they've shown is that if you ligate, if you cut those sympathetic nerve fibers right at the coronary artery, you cease progression of atherosclerosis. Very consistent with the liver and the pancreas studies. The brain, I would submit, is also involved. Kidneys. There are studies showing that if you, in models of chronic kidney disease, driven by hypertension, diabetes, in those models of kidney disease, if you cut the sympathetic fibers going to the kidneys, you stop the progression of that chronic kidney disease. Also, look at skeletal muscles. The same thing is going on with muscle. And this is largely, I believe, what's behind the sarcopenia of aging. And if you look in individuals who are obese, have diabetes, their muscle quality is low, and they develop a sarcopenia of obesity. This is what's going on also at the skeletal muscles. So I wanted to talk about a topic that very few people have heard about, but it's very much a part of diabetes, and it involves the liver and the pancreas, and what more and more researchers are calling the liver alpha cell axis. This is essentially a mechanism or a pathway in which the liver and the pancreas communicate with each other. It involves glucagon resistance. Traditional thinking is that a glucagon and insulin, they're antagonistic. Glucagon's mission is to increase blood glucose. Insulin's mission is to lower blood glucose. And we have this very oversimplified view of it. The truth is, it's far more complicated than that and nuanced. Glucagon's effect on blood glucose very much depends on the context. In a healthy person, if blood glucose is elevated, high glucagon will cause the secretion of more insulin to bring down blood glucose. A lot of people don't know that. If blood glucose is low, glucagon will stimulate the liver to secrete glucose, gluconeogenesis, glycogenolysis. Now, the primary stimulator of glucagon release from the alpha cells of the pancreas is amino acids in the blood after a meal. But of course, the autonomic nervous system is also a driver of alpha cell secretion of glucagon. So I won't go into that, but a good argument could be made that it's primarily the brain and the nervous system driving glucagon release or secretion from the alpha cells. So say you eat a meal, it's a mixed meal, meaning there's glucose, there's fatty acids, there are amino acids. Those amino acids are transported in the blood, they reach the pancreas, they stimulate receptors on the alpha cells. The main ones are alanine and arginine. Those amino acids are pretty potent stimulators of the release of glucagon. Now, glucagon is released in the blood, It also is the primary driver of the GLP-1 receptors in the beta cells. Another topic, but the glucagon is a hormone, travels in the blood, a short distance between the pancreatic veins and the portal vein and to the liver. And in the liver, the glucagon signals to the cells of the liver to take in, to transport into the liver cells, amino acids in the blood. Again, we've just eaten, the amino acid levels in the blood go up. This is a signal from the pancreas to the liver to take in amino acids in the liver and to process them. Some of them can be turned into glucose, gluconeogenesis. But this is a very important process, and what we find in obesity is that there's what's called glucagon resistance. The amino acids in the blood, they stimulate glucagon release from the alpha cells of the pancreas. Well, you get a downregulation of glucagon receptors in the liver. So there's less receptors for glucagon. The liver becomes resistant to glucagon. So the level of glucagon in the blood goes up, as does the level of amino acids. There's decent data to suggest that this precedes true insulin resistance. And this is more correlated with obesity than it is diabetes. The way I think about that is it's likely this glucagon resistance may precede insulin resistance and be one of the drivers of insulin resistance. So back to glucagon in the pancreas. There's a very complicated relationship between alpha cells and beta cells. Again, we've sort of oversimplified those two cell types. Alpha cells, they generate glucagon. Beta cells, they generate insulin, and these two hormones oppose each other. It's far more complicated than that. It turns out that glucagon, which is produced and secreted by the alpha cell, that glucagon, of course, it binds to glucagon receptors. It also binds GLP-1 receptors. And there are GLP-1 receptors on the beta cells. So this is glucagon functioning in a paracrine manner. In other words, almost like a neurotransmitter. The alpha cells releasing glucagon. That glucagon will bind to GLP-1 receptors on the beta cell. And if there's glucose there, it will cause increased secretion of insulin. A lot of people don't know this. People think that the GLP-1 that's released in the gut or secreted and broken down in the gut, they think that is the GLP-1 that's stimulating the GLP-1 receptors in the pancreas. It's not so. At least it's not in a healthy person. The GLP-1, which is binding to GLP-1 receptors on beta cells of the pancreas, that GLP-1 comes from the alpha cells. You see, glucagon comes from the same gene transcript as GLP-1. From the same gene, this peptide can be broken down in different ways, either to produce glucagon or GLP-1. And so the alpha cell is very sophisticated. It's very nuanced. So it generates glucagon. It can also generate GLP-1. And studies have shown that proper insulin secretion from the beta cells after a meal depends on glucagon. And it depends on GLP-1 from the alpha cell. So it's really sort of a closed system in the pancreas, in the islet, where the alpha cell is largely regulating the beta cell. And there's feedback between them. The other important point is that, we talk about diabetes and oral glucose tolerance tests, all these tests that involve just glucose. Well, it's very different when you talk about a mixed meal. So in a mixed meal, you've got glucose, theoretically, you've got protein or amino acids, and you've got fatty acids. That mixed meal results in a greater insulin release, and it has to do with this interplay between alpha and beta cells. There's been great work done on the neurological driver of alpha and beta cell differentiation, and what has been demonstrated is that the beta cells can de-differentiate. In other words, based on the influence of the alpha cells, the beta cells can sort of regress into a de-differentiated state, and then they can differentiate again to alpha cells. And so you see this in cross sections or in pathologic specimens of diabetics. There's more alpha cells than beta cells. And the general thought is that with this increased sympathetic activity to the islet, it's stimulating alpha cells in a way that causes this change in differentiation between alpha and beta cells. On the topic of GLP-1, Several studies have demonstrated very elegantly that there are at least three, I believe there's four different systems, semi-closed systems of GLP-1. So we know about the gut, gut hormones. So actually, they're gut neurotransmitters. Those enteroendocrine cells of the intestines, they generate GLP-1, and that GLP-1 crosses a very tiny distance and stimulates the GLP-1 receptors on vagal afferent nerve fibers, and then that GLP-1 is broken down very quickly. That's the sort of enteric GLP-1 system. There's also this GLP-1 system in the pancreas between alpha and beta cells that we just talked about. There's also a GLP-1 system in the brain. There are centers in the brain, including in the medulla, the hypothalamus, that produce GLP-1 from the same gene, and you have GLP-1 receptors there. So again, this is a semi-closed system in the brain. Other studies have shown that GLP-1 is used in the olfactory bulb. This is for olfaction, for sense of smell. And GLP-1 in the olfactory bulb actually stimulates food drive, food searching. So sort of a paradox there. We think of it as a satiety messenger. In the olfactory bulb seems to be the opposite. Again, these molecules are far more sophisticated than we may think. On the idea of pleiotropic effects of all of these molecules, insulin. We mentioned before that insulin secreted from the beta cells of the pancreas. That insulin binds to insulin receptors right there in the islet on the vagal afferent fibers, and then that message, that signal is transmitted through the vagus nerve up into the NTS and up into the brain. Very fascinating. The brain also generates insulin. It has its own insulin receptors, but there are several areas in the brain that also produce insulin. The choroid plexus, several centers in the brain that generate insulin. And some studies would suggest that the insulin system in the brain is somewhat independent from the peripheral insulin system that we're normally used to thinking about. So there's several lines of evidence that the brain generates insulin. You can find messenger RNA of insulin in the brain, C-peptide in the brain. Fascinating. The Human Protein Atlas, a great resource. You go there. Just an incredible resource. According to the Human Protein Atlas, several areas in the brain where insulin is generated. So to summarize, Type 2 diabetes in human beings is a diet-induced neurological disease. It is driven primarily by high-fat, high-sugar food intake. And of course, genes are a part of it. Genes, I would say, dictate a person's susceptibility to the effects of these hyperpalatable foods. But really what we see are changes in the afferent signaling between gut and the brain. That's the first thing that happens. You suppress that signaling, and the downstream effects, all the dominoes start falling into place with the shift in the autonomic nervous system and end organ dysfunction. And in diabetes, the increase in systemic insulin, the increase in glucose, those effects are downstream from so much that is going on in the brain and in the autonomic nervous system. You can follow this out, this physiology and this pathophysiology, into almost any organ. And what you find is the same thing, like the liver or the kidneys. It is this imbalance in the autonomic nervous system that we really should be talking about. The carbohydrate insulin model, it's so limited. I spent 10 years there defending it, advocating for it. It's an oversimplification, and it does not account for any of this other stuff. Now, the thing about it is that it prescribes a low carb way of life. I personally subscribe to that. I'm low carb, been keto for 10 years or so. I'm probably carnivore at this point. But that doesn't mean that carbohydrates are the driver of the disease. It just so happens that cutting out carbohydrates most effectively allows a person to avoid the high fat, high sugar foods, those foods that are combined, have that combination of sugar and fat. That's why. But we shouldn't let that mislead us into thinking that we've narrowed it down. We know exactly what causes diabetes and these other metabolic problems. We're lying to ourselves. There's a bigger picture, and it's very important for us to see this bigger picture and to understand it, to really get ahead of this. Thank you for joining me. Hopefully this was helpful. For those of you who held out and listened to the whole thing, congratulations. That's a lot of information. We'll have more videos like this discussing all kinds of different manifestations of metabolic disease, coronary artery disease. Thanks for joining us. We'll see you next time.